Hello, welcome to CNA's Opus, a late night session here. Just got done with the workout routine, so I'm really tired. I hope I can get through this. See what we got on today's agenda. What I got here. Oh, shit. I'm gonna try to make it through this. I'm kind of tired right now, but okay. So you take a look at this right here. All right, wait. Can you see it? See this? All right. This is this is tumor suppressant protein P53, and there is MDM2 right above it right there. And what that wants to do is it wants to bind to it and inhibit it and target it for ubiquitination. So to put a little ubiquitin group onto it, and then it'll get sucked in by the proteasome, and the proteasome will chop it all up and everything. But once it's a phosphate group is added to it, MDM2 does not bind to it at all, so it can't target it. It can't put the ubiquitin group onto it. If you look at the next one down here, this is beta-catenin. And what beta-catenin is doing is not really all that important. What's important here is you have two scaffold proteins right here, down here, two scaffold proteins, axin and adenomous polyposis coli, which is another scaffold protein, and adenomous polyposis coli mutations in that are heavily linked with familial forms of cancer, but that's not really on the agenda right now, so I'm going to stay clear of that, and that's marked in APC right there in the red. These scaffolding proteins, all they really do is hold proteins to it, like certain kinds of enzymes, and one of the enzymes that it's holding to it is glycogen synthase 3 beta, which is a serine threonine kinase, which means it'll add a phosphate group on the serine or threonine. Right underneath that is casein kinase 1 alpha, which is also a serine threonine kinase, and that will add phosphate. The first step is beta-catenin will go in there and bind to axin, and then the amino group right there will be going in this way towards the axin scaffolding protein. And the first step is the casein kinase 1 alpha protein will add a phosphate group to a serine, actually at position 45, which means if you count it up from the amino terminus, 45 amino acids in is a serine residue, and it'll add a phosphate group to it right there. That's the first step, and that'll excite the top kinase enzyme, glycogen synthase 3 beta, which will phosphorylate all these three other ones downstream. Now that doesn't add a ubiquitin group to it. That just cleaves it away, and there's 4 phosphate groups on it from position 45 on down towards the amino terminus. Then a protein called beta TRCP will bind to those phosphate groups there and add a ubiquitin group onto it. But what I want you to see here is that P53 does not get targeted for ubiquitination because a phosphate group was added to it. This beta-catenin is targeted for ubiquitination because phosphate groups are put onto it. So it's the exact opposite thing is happening right there. Now if we shift right down here, I don't know if you can see this, but this right here is hypoxia-inducible factor 1 alpha, which plays an important role in angiogenesis. But in normoxic conditions, in conditions where there's enough oxygen for the cell to survive and thrive, the HIF1 alpha is made and destroyed, just like beta-catenin and just like P53. They're proteins that are constantly being made in the cell and constantly being destroyed until they're needed. Until they're needed. So right here on hypoxia inducible factor 1 alpha, there's 2 prolines right here. See the 2 P's right there? And proline is a weird kind of amino acid because it forms a ring off the alpha carbon right here. I can't get my fingers towards the right spot. Right here, off the alpha carbon, it forms a ring. 1, 2, 3. 3 carbons off the alpha carbon back to the amino group. So the amino group doesn't have a hydrogen off of it at all. And that's what makes proline a little bit unusual. If you look down here, if I can get this board right here, and right there. So an enzyme, when it's in normoxic conditions, which means there's enough oxygen, you want HIF1 alpha destroyed, or hypoxia inducible factor 1 alpha destroyed. And propyl hydroxylase will bring over a hydroxyl group and put it on the 4th carbon. So it'll take off a hydrogen and put a hydroxyl group onto the 4th carbon. See, because it's 1, if you count from the carboxylic acid, it's 1, 2, 3, and then I can't get my fingers right because I'm looking backwards. 4, and that's the 4th carbon. So there's a hydroxyl group off of it, and we call that 4-hydroxyproline because it's a hydroxyl group off the 4th carbon. And then a tumor suppressant protein, von Hippel-Lindau. Von Hippel-Lindau, right there. Which mutations in this can cause a disorder called von Hippel-Lindau syndrome or disorder. And it's a tumor suppressant protein, but it adds the it will, as soon as this is hydroxylated on both of the prolines, the von Hippel-Lindau protein will bind to it and put a ubiquitin group onto it. Now, the important thing to see out of all of this, the point that I'm trying to get across, is that all of these here, how come I can't get this right here? All of these here, the ubiquitination process starts at a different way. This one gets ubiquinated when no phosphate groups are added at P53 right here. And if you don't know about P53, P53 is vital for error correction of DNA. It stops the cell cycle by producing tumor suppressant protein P21. It can produce PUMA and NOXA, which are both BH3 only proteins. And then down here, this is beta-catenin, which drives the cell cycle from interphase into G1 phase. And this down here is hypoxia inducible factor. When it's in hypoxic environment, which means it's low in oxygen, it doesn't get destroyed, and it'll bind with something in the nucleus called hypoxia inducible factor 1 beta, and it will start producing vascular endothelial growth factor A for angiogenesis. So all of these have vital roles in the cell, but they're constantly being made and constantly being destroyed. But the point that I want to get across is P53 is phosphorylated, and it doesn't get ubiquinized, and it doesn't get destroyed. Beta-catenin is phosphorylated, and therefore is ubiquitinized, and therefore is destroyed. And this one down here, hypoxia inducible factor 1 alpha, doesn't get phosphorylated at all. There's no phosphate groups put on it, and there's just a hydroxyl group put on it, and it still gets targeted for ubiquitination. So the point of all of this is that the ubiquitin, the proteasome ubiquitin pathway. It has a starting point that can be completely different for different proteins within the cell. So if you want to see it as kind of a dogma, the starting point of ubiquitination is different. That's the point I'm trying to get across. Is that the starting point for ubiquitination is different for different proteins within the cell. And that's what I'm trying to get to. And that's it. Rich, CNA, see you later. I hope next one I'm a little bit more awake. But I hope you got something out of that.